Hi team, welcome to SRL Bio. This is my take on practice paper 1 from the CGP white workbook. That's this one. We're going to go through question by question reasonably quickly, focusing on things like decoding the questions and making sure that we're covering everything that we possibly need to cover. Let's do it. Okay, so question one is focusing on human reproductive systems. We've got quite a lot of factual stuff in here. It's a nice sort of open goal to start with. This is all assessments at objective 1, that's the recall stuff. So let's chuck some answers in. A word that describes cells that contain half the usual number of chromosomes is haploid. How many chromosomes in one gamete in a human is 23, 46 in a diploid, that's a body cell, 12 and 32 are just nonsense. Which of the following structures in the human reproductive system is the site of gamete production? That's testes. This is in the male system, so the vas deferens is the sperm duct, so that's this one running up here at the top. The urethra is the tube coming down here, so from down out of the penis, and then the erectile tissue surrounds the urethra, so none of that, so testes. An embryo develops in the human female reproductive system. Describe how a human embryo is formed. Right, this is talking about fertilization, right? So firstly, we want to be adding the word fertilization. So we want sperm plus egg forms a zygote, and then the zygote, remember that's that single cell that results in the fertilization, so the fusion of the sperm and the egg cell. That zygote then has to undergo mitosis in order to form the embryo, which is that ball of cells. So the zygote undergoes mitosis to form the embryo. Okay, nice. That's question 1. Okay, so question 2 is a photosynthesis one. I'm focusing on the practical aspects of that. So there's a couple of spec practicals in here to get your teeth into. So this setup is a reasonably straightforward one. We've got some pondweed, we've got it underwater, there's the measuring cylinder on top of that, and we're collecting the gas into there so we can see the volume of the gas being given off, and then we've got our lamp providing light for photosynthesis. So the gas being collected in the measuring cylinder is oxygen. That is the waste product of photosynthesis. Explaining what would happen to the volume of gas collected if the investigation was repeated with the lamp turned off. We've got 3 marks here, right? So that's where my brain is first going here. With an explain question, we're going to have to be saying why. And for 3 marks, this is looking for a little bit of detail. So we'll start with lamp turned off, so we're getting less light. For a start, that's going to give us an entry point into photosynthesis here. So less light means less photosynthesis. And if we've got less photosynthesis, then we're going to get less oxygen. So oxygen is a product of photosynthesis, and therefore less photosynthesis means less oxygen. So oxygen is a product of photosynthesis. Therefore, less photosynthesis will give you a lower volume of oxygen. Okay, sodium hydrogen carbonate dissolves in water and releases carbon dioxide. Suggests why sodium hydrogen carbonate was added to the water in this experiment. Well, again, this is looking at the requirements and products of photosynthesis. So when it dissolves in water, it's providing carbon dioxide. Carbon dioxide is a requirement of photosynthesis. So again, we're looking down here, we've got 2 marks. Our brains have got to be going here into what those 2 marks are going to be for. So they haven't told us in the question that carbon dioxide is a requirement of photosynthesis. That's a hint that there's going to be a mark for that in here. So carbon dioxide is required for photosynthesis. It's just why sodium hydrogen carbonate was added to the water in this experiment. Yes, carbon dioxide is a requirement for photosynthesis. There would be some carbon dioxide in the water anyway. The reason that we'd want to add this to the experiment is so that carbon dioxide is not limiting. We're trying to investigate how the amount of light available is affecting the rate of photosynthesis. What we don't want, therefore, is for the concentration of carbon dioxide to start limiting that halfway through. for whatever reason. It gets used up in the water. So we want carbon dioxide in that water in excess, so that it's always the light that is limiting the rate of photosynthesis, or as much as possible, it's the light that's limiting the rate of photosynthesis to give us the best possible answer when we're doing our experiment. So carbon dioxide being required for photosynthesis would be our first mark here, and then the second mark here would be that adding sodium, hydrogen, carbonate means carbon dioxide is not, or concentration of carbon dioxide I should say, is not limiting the rate of photosynthesis. Okay, onwards to part D. Explain how temperature affects the rate of photosynthesis and suggest how temperature could be controlled in this experiment. Okay, this is the first point here where we'll really ram home this idea of decoding questions and spotting questions to which you know the answer, right? In just about every paper one, you're going to at some point encounter this idea of an enzyme, probably temperature, it could be other enzyme limiting factor questions, but if you can spot the question that runs through enzymes and temperature, that's, well, in this case, 3 marks that are straight off the bat. So in this case, we've got explain how temperature, as soon as you see temperature there, your brain should be going to enzymes. This could easily be a 5-mark question, right? Explaining how temperature affects the rate of any reaction, you can rattle off at least 4 or 5 marking points about that. We'll just quickly do that. So as temperature increases, that means we have more kinetic energy. And therefore more collisions between enzymes and substrates. More collisions, therefore a faster reaction. Okay, so that's the first half of our temperature answer. The second half is beyond or above the optimum temperature. Enzymes will denature. Substrates no longer fit in the active site. So the reaction stops. Now there's some other little marking points that you could get in there, things like what denaturing is, so that denaturing being a permanent change in the shape of the active site. You could also talk about how if the substrate no longer fits in the active site, then you're not forming any enzyme substrate complexes, possibly for another mark. I'm going to stop there just because this is a 3-mark question, but that could easily be 4, 5, 6 steps you can put in there. And the second half of this question suggests how temperature can be controlled in an experiment. Anytime you want to be controlling temperature, we go water bath. So the second half of this, we'll just say control with a water bath. All right, the temptation here, or the most common error, is to be talking about controlling temperature using a thermometer. That's not the case. You can't control temperature with a thermometer. A thermometer tells you what temperature it is, but it doesn't keep that temperature the same. Water bath. Okay, E. Which of the following is not a stage in testing a leaf for starch? So this is another spec practical. This is the one where you bleach the leaf and then you test it with iodine to see where the starch is. So boiling the leaf to stop any chemical reactions happening, we do that in the first place. So that's the first thing we'll do, is boil the leaf. That stops the chemical reactions. It also removes the waxy cuticle if it's got one. Drying the leaf in the oven to remove water, we don't need to do that. So this is going to be B. Heating the leaf with ethanol to remove chlorophyll is the case. We do need to do that. That bleaches the leaf and turns it white. That means that when we drop the iodine on it, you get a nice clear result. And the iodine itself is option D. Okay, would you expect the test to produce a positive result? Explain your answer. Well, yes, because clearly there is photosynthesis happening here. We know that because we've got oxygen given off. And so that glucose that's being produced from photosynthesis is being stored as starch. So yes, we can say photosynthesis produces glucose, which is stored as starch. You'll notice I'm being reasonably notey here. You probably want to be writing a little bit more prosily than I am here, but just to get the key points in, I'll try and break it up a little bit. Okay, that's question 2. On to question 3. Okay, so here's question 3. Question 3 is set itself up here as kind of a digestion question with a little bit of red blood cells thrown in at the end, but actually, if you look through this, this is just diffusion, right? And quite simple diffusion at that. So we've got 2 structures, villi, and then we look at red blood cells, but both of them are just coming down to diffusion and that theory behind maximizing the rate of diffusion across an exchange surface. So this is true of villi, it's true of the alveoli and the lungs, it's true of any surface across which diffusion is happening. So 3 ways that villi are adapted to absorb the products of digestion. Now you could basically do this just from your general kind of exchange surface maximizing diffusion theory. So if we were just to jot that down, it would be something like a large surface area to volume ratio. We'd have a good blood supply. We'd have a short diffusion pathway. And those 3 things combined then would lead to an effective exchange surface. Now, to be specific to a villus, we might want to expand on this large surface area to volume point. So we could talk about the microvilli. So those are the projections on the individual cells that make up the lining of the villus, so the single layer around the outside. The good blood supply, that's obviously capillaries. So we've got a capillary bed within the villus. The short diffusion pathway, so this is the wall of the villus is 1 cell thick. I'm pretty convinced here we've already got full marks. We could add in the one specific point to a villus, which would be the lacteal. So the lacteal is the projection that goes up through the middle of the villus. It's involved with absorbing the products of fat digestion. So we'll chuck that in there because that is still an adaptation to absorb the products of digestion, even though in itself the lacteal is not necessarily linked with diffusion and increasing the rate of diffusion specifically. So the lacteal involved with lipid absorption. Okay, one thing to point out on here, I guess, the short diffusion pathway, this is where one of our most common, I wouldn't say misconceptions, but errors in phrasing comes in. It's here when we say 1 cell thick and the wall of the villus. This comes up over and over again. So whether it's a villus or an alveolus, is to avoid the phrasing cell wall. When we're talking about exchange surfaces, if we talk about an exchange surface having a thin wall, or the wall of it being 1 cell thick, that's the distance that something has to diffuse to get from one side to the other. These are animals, so there are no cell walls. So just be careful to avoid the phrasing cell wall within a description of any animal structure. Okay, on we go. Diagram shows glucose being absorbed into the blood by diffusion. Describe how amino acids are absorbed into the blood. Okay, so this next part is asking about the absorption of amino acids into the blood. Here we just need a little bit of knowledge. Amino acids are absorbed by active transport. So that will get us our first mark. So amino acids are absorbed by active transport. Okay, now the second mark in here, right, this is a 2 marker, there's going to be a mark here for some sort of description of active transport itself. So something about what makes active transport different from diffusion. So we'll say something like active transport requires energy, or ATP, and moves amino acids against their concentration gradient. and that will get us our second marker there. Okay, part C, diagram shows red blood cells in the blood. So that's the diagram at the top of the question. State the function of red blood cells, right? 1 mark, red blood cells, transport of oxygen from the lungs to tissues. It's probably better, isn't it? There we go. Explain how red blood cells are adapted to their function. Okay, again, this is a bit of knowledge, but equally, if you've got an understanding here that we need oxygen to diffuse in and out of a red blood cell, then we can just go back to our answer to part A and just readapt that for red blood cells, and we're picking up marks again. Right, the large surface area to volume thing, that still applies. We've just got different structures underpinning that now. So how does a red blood cell get a large surface area to volume ratio? Well, it's small. That always gives something a high surface area to volume ratio, and it's got a biconcave shape, so that biconcave disc that increases the surface area. We also have, and this is where we're just talking knowledge here now, right? So hemoglobin in the red blood cells carries oxygen, and the fact that there's no nucleus, which leaves more space for hemoglobin. Last point in here, I suppose, is flexibility. So red blood cells can fit and move through thin capillaries. All right, that's quite important. The width of a capillary is almost exactly the same as the width of a red blood cell. So they need to be a little bit flexible just so they don't get stuck and cause blockages within the capillaries as they move through. Okay, 3 marks in there. I reckon we've probably got 5 marking points. The size, the biconcave shape, the hemoglobin, the fact that they don't have a nucleus, and the fact that they're flexible. 5 decent adaptations of a red blood cell. That'll do for question 3. On we go. Okay, so question 4 is on plant responses and phototropism, to be specific. So we've got a student growing 3 plants in a tray. That's diagram 1. Then they put that in a cardboard box with a cutout hole in the corner, and that's shown in diagram 2. Draw a diagram to show how you'd expect the plants in the box to look after 3 days. Okay, so this is just testing our knowledge of phototropism and the fact that any plant, when grown with a directional light source, will grow towards that light. Plants need light for photosynthesis, so very simple and straightforward evolutionary adaptation to grow towards it. So we've got to do some sketching. So we're going to need our box with the cutout hole. And then we've got our tray down the bottom and our shoots, which would have grown towards that light. Something like that. Put some leaves on there for good measure. Right, explain what caused the plant's response. First thing we're going to do with an explain question, as with any extended response ones, have a look down here, 4 marks. All right, so to get 4 marks out of here, we're going to need to be quite detailed, and we're going to need to be quite specific in what we're saying to pick up all 4. So we'll run through this step by step. First thing we've got is a directional light source. So light is coming in through the hole in the box. So light only coming in through the hole in the box. That's going to cause a shift in the auxin distribution within the plants. Remember, auxin is that plant hormone which causes shoot cells to elongate and root cells to, or restricts the elongation of root cells. So that causes auxin to accumulate. So auxin accumulates on the shaded side of the shoots. Cells on the shaded side elongate. The shoot bends towards the light source. Nice, there you go, 4 points, 4 marks. Which of the following responses is shown by the plants in this experiment? Positive phototropism is that one. right? So we talked about this being phototropism. It's photo means light, tropism means turning. So a positive phototropism means you are turning towards the light source. Negative phototropism would be the opposite of that, so turning away from the light source. Geotropism refers to the earth and gravity. So positive geotropism is turning towards gravity. Negative geotropism would be turning away from gravity. Very good, there you go. Question 4. Okay, so question 5 is our first dive into genetics in this paper. So we're talking about cystic fibrosis. This is a really common example. This is a genetic disorder. It's a recessive genetic disorder. We have a couple that have a baby boy. Doctor tells them the baby has inherited cystic fibrosis. Neither parent shows signs of the disorder. So we've got 2 parents that don't have this recessive condition and an offspring that does. In the space below, construct a diagram to show how the baby inherited cystic fibrosis. Now, there's a couple of different diagrams that you can legitimately draw here. I would always encourage you to draw the Punnett square. This is a straightforward diagram. It's very difficult to make sort of simple errors with it, and it's very clear to the examiner that you know what you're talking about. So we will draw a Punnett square and we'll pick up 3 marks from that. So diagram shows genotypes of the parents, genotypes of the gametes, all possible genotypes of the offspring, so we don't need to show any phenotypes in this. Quite often these are 4-mark questions in which you do need to give phenotypes, in which case my best advice is to draw the Punnett square big enough that you can fit the phenotypes into it, and that leaves no room for misallocating phenotypes to different genotypes, and in almost all cases that will pick up the mark. You just need to make sure that you read these questions carefully and make sure that you've answered every part of that question. However many marks there will be a list in the question, it's here for this one, that tells you exactly what the examiner is looking for in order to get those marks. So we know here that we've got an offspring showing a recessive condition that neither parent does. That means that both parents must be heterozygous. So they must carry both the dominant allele, which means they don't have the condition, but also carry the recessive allele. So the offspring can inherit both of those recessive alleles and then have the condition. So my Punnett square will look something like this. I'll put the 1 parent up there, 2nd parent down the side, and then my offspring, I have homozygous dominant, heterozygous, and homozygous recessive in the bottom right. This then bottom right, this is my individual with cystic fibrosis. So that's the offspring. That's the baby boy in this case. Is the baby homozygous or heterozygous for this condition? He's homozygous. And explain my answer is because the condition is recessive. So a recessive condition, you can only show a recessive condition if you are homozygous recessive. Okay, the doctor tells the parents if they have another child, fetus can be tested to see if they have cystic fibrosis. State the probability that the couple's next baby will have cystic fibrosis. Okay, so this is testing our knowledge of this Punnett square at the top that we've already drawn. So that's giving us a 1 in 4 probability. But also just to be clear that the probabilities don't change with every offspring. So it's not the case here that because we've had an offspring with cystic fibrosis, that the next 3, for example, wouldn't, to make it up to 1 in 4. Each individual offspring has a 1 in 4 probability. So on average, if you have 4 children, the average number with cystic fibrosis from this couple would be 1, but that doesn't apply at an individual level. So it's still 1 in 4, or 0.25. Would be the probability in there. Okay, heading to the other side then, we have our pedigree. We've got a family here also with cystic fibrosis. We're being shown here the carriers. We're not always shown carriers. This is a genotypic tree, so that it shows us it shows us the heterozygotes. You can also have phenotypic trees where the heterozygotes and the homozygous dominant individuals would just be shown the same. In this case, they'd be shown as non-sufferers. Okay, so we want to explain what Lena's genotype must be. For that, we need to look at her whole family, right? So we've got Lena in here. She's married to Ian, or is having offspring with Ian. And they have 3 kids, Karis, Beth, and Alfie. Karis is an unaffected female. Beth has cystic fibrosis, and Alfie is a carrier. Now, in order to have all 3 of those conditions, homozygous dominant, homozygous recessive, and heterozygous, the only way that you can have that is if you have 2 heterozygous parents. You can't have that combination of offspring with any other parental combination. If Lena was unaffected, so homozygous unaffected, then all of the offspring would have to inherit that dominant allele from her, and she wouldn't have any offspring showing cystic fibrosis. If she had cystic fibrosis, then you wouldn't be able to have a Caris in this case. So you wouldn't be able to have the unaffected offspring if she had cystic fibrosis, because all her offspring would inherit that recessive allele from her, and so they'd at least be heterozygous. So Caris would be impossible. So in this case, she must be a heterozygous female. So she must be heterozygous because otherwise she couldn't have offspring of big F, little f, and heterozygous as well. Okay, so you couldn't have all three if you're not heterozygous in both parents. Caris and Beth are sisters. Caris has a scar on her hand. Beth does not. Explain whether this is an example of genetic, environmental variation, or a combination of both. That's environmental. So a scar is not caused by genes, but by experiences during Caris's lifetime. Okay, so any scar is obviously going to be a result of damage to the skin during someone's life. You're not going to get born with a scar like that. Okay, and that's question 5. Okay, so question 6 kicks off then with natural selection and a pretty sort of model case study on natural selection. This is the peppered moth. I'm sure you've come across peppered moths in the various case studies of this you've looked at. This is a moth which has 2 colour morphs, so there's a light variety and a dark variety, and the transition from the light to the dark, or a light population to a dark population, was shown during the Industrial Revolution, where trees got covered in soot. And so that's what this question is introducing. And then also using the idea of natural selection, explain whether the dark variety of moth became more common in soot polluted areas. Okay, so 4 marks for this. This is natural selection model answer territory. All right, if we've got a natural selection model answer to reel off, then this question doesn't take very much in the way of decoding. We just insert our case study. With any natural selection model answer, we start with mutation. So mutation creates variation in colour in the population. Okay, now that might be going back a little bit too far for this question because it's already talking about the dark variety in the question, but we'll start with it. This is our model answer. So mutation creates variation in colour. Once you've got that variation, then we've got an environmental change. So the trees become darker as they're covered in soot. At that point, we're changing the environmental selection pressures. So the idea here that now the white moths are more likely to be predated because the black ones have better camouflage. So we'll say the black moths are now camouflaged, and so the white ones are more likely to be predated. Okay, and then we're heading through our natural selection answer into survival and reproduction. So that means that our black moths are more likely to survive and reproduce. That means then that our black alleles, so the alleles that are responsible for the black coloration, are more likely to be passed on to the next generation, more likely to be passed on, and then this just repeats over generations. Now it's hard to say in a question like this whether it's 4 marks. This could be a 5 marker. It's hard to say exactly which of those they'll be looking for in a mark scheme. Certainly, I would suggest here that the survival and reproduction of the black moths because of predation of the white ones is probably going to be our key. So we're probably looking at these points here as being the key ones for that mark scheme. But it's worth just having natural selection as a model answer just to rattle off so that you can apply any case study that they throw at you to it. Okay, so the bar charts on the right hand side, we've got the percentages of dark and light coloured. This is in two different places. We've got town A, town B. Which one's the most polluted? Well, the most polluted one is likely to be the one where we've got more of the dark variety because they are camouflaged, they're better adapted. So B will be most polluted. Give a reason for your answer. Dark variety is better adapted to polluted areas because it is camouflaged. Okay, part C, bit of maths. Calculate the difference in percentage between the dark coloured moth population in town A, town B. Show your working. Okay, difference in percentage. This would be easily confused with the percentage difference. Because this is percentage data, the percentage of a percentage just gets a little bit kind of meta. So actually what we're looking at here is just a straightforward subtraction. We want to know the difference between this percentage, when we read that off there, and this percentage, when we read that off there. This one is 25 and this one is 70, it's just above that line, so that's 77. So it's just 77 minus 25 and 52% therefore for our 2 marks in there. Okay, that's question 6, on to 7. Okay, so question 7 is an osmosis one, but largely some data handling really is getting you the bulk of the marks at here. So this is, again, a spec practical. We've got potato cylinders in different concentrations of a sugar solution, and we're looking at the change of mass of those potato cylinders as a result of osmosis in and out of those cells. So in different concentrations, there'll be different differences in water potential and therefore different changes in mass. So in the distilled water, so at zero concentration of 0 sugar, we've got a significant increase in mass, and that increase in mass decreases and decreases and decreases down to the most concentrated sugar solution where the potato is losing quite a lot of mass. Okay, so part A, calculate the mean change in the 1.2 mole of sugar solution, show your working. So that's these 3 data points here. We want an average of those. So we're going to add them all up and divide by 3. If we add all of those up and divide by 3, what did it to? Okay, to 2 decimal places is going to be minus 0.8. 0. Okay, draw a graph of the concentrated sugar solution against the mean change of mass in the grid below. Use straight lines to join your points. Okay, now just as a forewarning, in your exam, they won't draw axes on like this for you. You will just get a sheet of graph paper. And in actual fact, I think this makes us a little bit more awkward because we've got positive and negative data that we want to show on the y-axis. And so our x-intercept is going to be a little bit weird here, but we'll draw it nonetheless. Okay. So we want our independent variable along the x-axis and our dependent variable along the y. So independent variable is our concentration of sugar solution, and our unit is molar. And up the side. Okay, so we set our axes up like this. That's going to encompass all of our data. Now we've just got to plot those points on there straight off this graph on the left-hand side, table on the left-hand side. And because this is an exam, we're going to do this properly. Okay, it's not the best bit of line drawing you'll see. I'm sure you could do better on paper, but point is we are going straight lines and we're going point to point, particularly when it says in the instructions, use straight lines to join the points. If they don't say anything, go dot to dot, with a ruler. Okay, the only thing we haven't done is to label this up here. So this is the mean change in mass, which is grams. Estimate the concentration of sugar inside the original potatoes. Explain your answer. Okay, our estimation here is it's going to be 0.4 because that's the point where there is no change in mass. That tells us the water potential is the same on the inside of the potato cells as it is in the solution in which those cells are bathed. So this is just going to be a read off the, well, we don't really need to read it off the graph because it's in our results table, but we'll say 0.4 because there is no change in mass of the potatoes. Potato cylinders. Okay, suggest why the student used 3 potato cylinders in each concentration sugar solution to mean of the results. This is repetition of an experiment, and repetition is about reliability. So this makes the results more reliable. There we go. That's question 7. Okay, so question 8 is our dive into ecology. So, diagram shows the amount of energy contained within an area of plants and how much of that energy is transferred to each trophic level within the food chain. Plants are the producers, which the following statement describes a producer, the first consumer in a food chain. Well, producer and a consumer are 2 different things. So no organism gets its food from other organisms. That's not true. That would be a consumer. An organism that gets eaten by tertiary consumers, or tertiary consumers will eat secondary consumers. So again, that's not a producer. An organism that makes its own food using energy from the sun, that is a producer. So that's D. State how much energy is available to the tertiary consumers in this food chain. Okay, so the tertiary consumers we've got in this food chain is our producer, our primary consumer, our secondary consumer, and our tertiary consumer. So our tertiary consumer are the snakes. The energy available to the snakes then is whatever's in the mice, so that's 130 joules. Okay, the population of bluebirds increases. Bluebirds feed on the grasshoppers, causing the population of grasshoppers to decrease. Explain one way the change in grasshopper population might affect the other organisms in this food chain. Explain your answer. Okay, so there's essentially two ways you can go with this. You can either say that the plants will increase because fewer grasshoppers to eat them, or you can say that the mice will decrease because there are fewer grasshoppers to eat. So either you're looking up the food chain or down it. Either one of those would get you your 2 marks. Okay, 4 pyramids of biomass are shown, A, B, C, and D. Explain which pyramid of biomass represents this food chain. Okay, now we've got a little bit of iffiness in this question in that so far we've been given energy data and now we're being asked about biomass. And those two are not entirely the same thing. That being said, it's going to be D because the energy density between these organisms is not going to overcome the fact that we're going 11,000, 1,100, 130, and 12. So we're losing about 90% of the energy at each stage here. So it's going to be D because biomass decreases at each trophic level. And realistically, any pyramid of biomass for any ecosystem is going to have that shape in general. Okay, calculate the percentage of energy in the grasshoppers that is transferred to the mice. Show you're working. Okay, so this is 130 joules that ends up in the mice out of 1,100, which is the amount of energy that's in the grasshoppers, and we multiply that by 100, and that gives us 11.8% according to my calculator. Okay, the food chain has 4 trophic levels. Most food chains have no more than 5 trophic levels, just why the length of food chain is limited in this way. This is about energy loss. So we can say energy is lost at each trophic level. So there's not enough energy to support trophic levels above the 5th. You can find very rare instances of 6, maybe 7 trophic levels, but realistically that is limiting, or that is limited by the energy in the system. Okay, that's question 8. Okay, so question 9 is lungs and breathing. Right, we have gases A and B diffusing in and out of the blood. A is going into the blood and is therefore oxygen. B is coming out of the blood plasma and is therefore carbon dioxide. Okay, 3 ways that alveoli are adaptive to gas exchange. Now, we've been through this because we've already done a diffusion question. So ultimately, we're looking at the same kinds of things that we talked about with the villi in the earlier question. So we have, for example, large surface area to volume ratio. We have the blood supply, the capillaries. We have thin walls, brackets 1 cell thick. Again, we're just avoiding that phrasing of cell wall in there. And there we go, there's our 3 marks. The other things you can have in there, the moist lining as well as specific to alveoli. So the moist lining will help gases to diffuse. Gases diffuse much better in a dissolved state. So they dissolve in that moist lining and then they'll diffuse across into the cells. Okay, ventilation allows air to enter and leave the lungs. Describe the process that results in air leaving the lungs. Okay, so this is breathing out. We need to be talking about, with any ventilation question, we need to be talking about what muscles are contracting and relaxing, what then happens to the pressure within the lungs, and therefore what happens to the air. So in this case, air is leaving the lungs, this is breathing out. So we'll start this with the muscles. So the intercostal muscles and the diaphragm will relax. That causes the rib cage to move down and in, and so the volume of the thorax decreases. That means the pressure in there is going to increase, and therefore the air is forced out. Good, and that will do it for question 9. Okay, so question 10 is our Corms question. So we're going to make sure that this is set out in prose and that it's well written, but nonetheless use Corms as a structure to base our answer around. So the way that I would encourage this is to annotate this off to the side. So we will work out what the Corms mark scheme is going to be, and then we will write our experiment and make sure that we hit all of those points. So C has changed, that's our independent variable. We're going to grow some in and some out of a tunnel. So this is just annotation on the right hand side. The O is the organism, so we need to have some sort of control variable about the organism. So in this case, maybe we'll go for the same species of lettuce. R is repeats, so we need to make sure that we are remembering to repeat our experiment. The 2 M marks here, this is a growth rate, so there is a time element to this. So I'd expect one of the M marks to be to leave these for a stated time period, and then for the second one to be talking about how we're going to measure growth itself. So what is it that we're actually going to measure, and after how long are we going to measure it? So in this case, we'll go for measure the mass of the lettuce, and let's say after I don't know, 3 months. As long as we've got a sensible suggestion of a time period in there, we'll get the mark for it, whatever it is. Okay, and then 2 S marks. So we want, these are control variables, so we need to be thinking about what we're going to keep the same between the 2 tunnels. So what could influence growth rate of a plant, which we could control between the 2. So we could have light levels if we can. We could control watering. We could control the soil. So the mineral content. Possibly the soil pH we might be able to control. Maybe fertilizer in there as well. Okay, that will probably do for us. We've got a decent number of control variables in there. Okay, so now I'm going to take that and we're going to convert it into an actual method. This needs to be an experimental method that we could follow, even though it is a Corms answer. We're going to place 50, just pluck a number out there, lots of repeats, 50 lettuce seedlings in a polytunnel and 50 outside. Now actually what I wanted to do first there was weigh them, so we'll just add that to the end of that sentence. So having first measured masses. Then we're going to leave for 3 months, ensuring that both sets receive equal watering, fertilizer, light, and are grown in the same soil. Okay, and then we're going to measure mass of each plant after 3 months. The 50 plants there is my repetition, right? I've got 50 individuals that I'm dealing with. The more individuals you put down there, the more repeats you're doing. So that's fine. We're then going to average the masses. Average the changes in mass. Now, realistically, probably here you'd be wanting to do a percentage change. So the bigger seedlings you'd expect to grow that much bigger. But we don't need to go into that kind of level of mathematical analysis in this. Corms questions don't tend to give marks for what you're going to do after you've got your results. So you won't get marks for saying things like draw a graph of the result or calculate percentage differences, things like that. They're just interested in what you're going to do in the experiment. How do you get your results? Then we're going to go back to my Corms notes and make sure that we've ticked everything off. Now we haven't here because we haven't talked about the species of lettuce, so we need to add that in. So in the first sentence, place 50 lettuce seedlings in a polytunnel and 50 outside, having first of their masses. We could probably just add a little note in there, all of the same species. That will get us our O marks. R mark, we've talked about the repeats because we've got 50 in each. The M marks, we've measured the mass before, we've measured the mass after, and we've measured the mass, we've stated the time period, so 3 months. And then my S marks in there, I've dealt with in my second bullet point. Okay, so I've got my notes for my Corms answer. I've written that out as an actual method, and then I've gone back to my Corms notes to make sure that I've ticked off every point that I've made. That makes sure that I am giving an actual experimental method, which I need to do for a Corms answer, but also that I'm appreciating what those marks are going to be there for and what I'm going to be picking up and making sure that I'm getting 6 out of 6. Okay, there we go. That's the Corms. On we go. Okay, so question 11 is looking at mineral requirements in plants. So a little niche bit of spec point here, but magnesium and nitrates, which are referred to in this question, are both little bits of knowledge that we need. And questions A and B are going to test that knowledge of what those substances are used for. So we've got 3 beakers. We're measuring heights of pea plants and the changes in height. And then we're being asked to explain the results, essentially. So explain why the growth in beaker B was poor. Beaker B has low nitrates. So nitrates are needed to make amino acids which are needed to make proteins for growth. Therefore, B, which was lacking nitrates, grew slowly or grew less. Okay, so there we go. We've talked about the fact that B is lacking nitrates, so we've linked that back to our information in the question, and we've told the examiner what nitrates are needed for, and then we've linked that to the growth. Okay, B, describe how you'd expect the pea plant in beaker C, which is low in magnesium, to look at the end of the week. All right, so magnesium is required to make chlorophyll. So the first thing you'll see in a plant which is lacking magnesium is yellowing of the leaves. After that, you'll start to see more stunted growth because it can't photosynthesize as well, so it can't generate enough glucose, so it doesn't have enough energy to grow. But the first thing you'll see, and about the end of a week is about right, you'll see a yellowing of the leaves. So we'll go for yellow leaves, low magnesium, which is required to produce chlorophyll. Okay, 2 factors the student would have to keep the same in each beaker to make the experiment a fair test. Right, so we're looking for control variables here. The one little underarm that generally is true. Temperature affects all reactions in biology. We know that because of the enzyme theory we talked about before. So if temperature is not an independent or dependent variable and has not been mentioned so far in the question, it will be a control variable. All right, so the first one we'll always put down here is temperature. After that, this is a plant growth thing. My leaning here would be towards photosynthesis. So something that's going to affect photosynthesis that we need to make sure is the same between all of them that hasn't already been mentioned. Light would be the most obvious. You could probably also have water, but we'll go for light because that's the most straightforward photosynthesis limiting factor. Okay, there you go. That's question 11. Okay, on with question 12. Question 12 is focusing on, we've got some data at the beginning here on smoking and coronary heart disease, and then we'll end up with some smoking impact on the lungs as well. Okay, so conclusions from the graph. 3 major conclusions we can look at here, right? The first is just the general trend, that there is an increase in incidence of death from coronary heart disease in those who smoke compared with those who don't smoke. So that's number 1. Okay, second point is then to be comparing the never smoked and the former smokers. So former smokers are less likely to die from coronary heart disease than those who do smoke. So if you give up smoking, you reduce your risk. That's one point. The other point then is that those who have never smoked have the lowest risk. Okay, scientists hypothesize anyone who smokes is more likely to die from coronary heart disease. Just one change you can make to the study to test the hypothesis. Okay, so the limitation of this study was 2 major limitations. One that it's only on men and one that it's on doctors, all right? So male British doctors. The British, you could possibly broaden out from British, but we'll focus on the male and the doctors things. So firstly, we need to include females because males and females might respond differently to these things. And secondly, that we're only looking at doctors. Now, doctors is not a particularly accurate cross-section of society. So in order to include or in order to hypothesize or test the hypothesis that everyone reacts in the same way, we'd need to broaden out that profession. So either include females or include other professions. Okay, 2 effects of smoking on the lungs. Now, these are pretty widespread. There's a lot of different effects that you could be talking about here. To get 4 marks, though, we need to make sure that we have both described and explained 2 effects. So start with a description of it and then explain why that effect is happening on the lungs based on smoking. Okay, so firstly, we'll make a little list of these, right? So ignore the 1 and 2 here because I will make a list. Right, damages the alveoli is going to be my first point. That can go as far as emphysema, where we have that breakdown of the alveolar walls within the bundle. So that can lead to emphysema. So that would be my description. Right, I've described an effect here. Emphysema is a description of an effect that smoking can have on the lungs. I then need to explain that effect, which is going to be reducing my surface area. So the explanation here is a reduced surface area that can reduce the gas exchange. Okay, so that'll be one. My next one, we'll go with tar. So tar damages the cilia. That will lead to a mucus buildup because the cilia are responsible for removing the mucus. And that will then lead to coughing because you need to cough then to remove the mucus that you can't remove naturally through the cilia beating. That coughing then, that's often referred to as a smoker's cough, and that cough will then irritate the lining of the lungs. So it'll irritate the bronchi and the bronchioles. That irritation will then manifest in well, it'll lead to infection and bronchitis in that swelling that the irritation will cause. Okay, so that would be my second point. The tar damaging the cilia, that build-up of mucus, the build-up of mucus causing a cough, coughing irritating the lining of the airways, and that might lead to infection and bronchitis being that swelling. Okay, the other point we'll make here is about cancer. So cigarette smoke contains a lot of carcinogens. Carcinogens will cause cancer. Okay, there we go, there's question 12. Okay, it's question 13. Question 13 is an experimental one. We've got a graph at the top here. This is an exercise and heart rate. This student is doing some exercise. They've measured their heart rate before they do the exercise, and then during, as they finish, and then they've got, not continuous, but a few measurements after they finish the exercise back to when their heart rate goes down to its resting rate. So the independent variable here, now this is a slightly tricky one. Our default when we look at experiments like this and you've got a graph is that your independent variable would be whatever's on the x-axis. Now in this case, you could make an argument for time being the independent variable. That is the thing that's changing and we're measuring our heart rate over time. However, the fact that we've changed the intensity of exercise, really that's what we're looking at here. You could very easily have a graph that showed this experiment with a bar chart. So your heart rate during exercise and your heart rate after exercise wouldn't give us as much detail, but actually that's what this experiment is showing you. So the independent variable here is not time, it's the intensity of exercise or the rate of exercise, or even exercise versus no exercise. But we'll phrase that as the intensity of exercise. Okay, using the graph to estimate the student's heart rate after 90 seconds, so we're just reading off the graph here. Again, in an exam we'd do this slightly more carefully, I suppose, but 90 seconds is halfway between 1 and 2 minutes, so we want halfway between these two. If you want to get really good, we'd be measuring that, and then we're reading horizontally off the graph to here. That's 128 or so, isn't it? There'll be a margin for error in the mark scheme there, particularly as this graph doesn't have any small divisions on it. There's a little bit of estimation in there. Okay, students stopped running 2 minutes after taking the first heart rate measurement. How long did it take for their heart rate to return to normal after they stopped running? So we're looking for the first normal value, which is here at 8 minutes. They stopped running after 2 minutes, and so we're going back from 8 to 2, so that's 6 minutes. So 8 minus 2 is 6 minutes. Make sure we include the unit in there. The unit's not given in the answer space, so we need to give it. Okay, explain how the student's heart rate changes with exercise. Now this is a slightly trickily worded one. The key here is in the word how. If you're explaining how something is happening, we want to be talking through the process from start to finish. The temptation here is to go down a sort of an oxygen route, and that nice classic answer about having less oxygen in the tissues and therefore increasing heart rate. But actually, we need to put some detail in there. So we've got to get the stages in here between the beginning of the exercise and the increase in the heart rate. So first thing, we have the word changes in here. because that's not directional, that's a bit of a clue that there might well be a mark here for saying what happens. So just a straightforward heart rate increases with exercise. Now that might well come at the end of that answer, but we'll get the easy mark first. Okay, next point in here is that the muscle tissue is respiring more, right? That's the underpinning all of this. Now, in order for that to lead to a higher heart rate, we need to be focusing on the carbon dioxide side of things. So this is actually a stimulus response chain. What's happening in your body here is that the receptors in your body are detecting a high carbon dioxide level. They're actually detecting a slight drop in the pH of your blood because as the carbon dioxide dissolves, it forms carbonic acid. So as your blood pH drops, those receptors, so there's one in your carotid artery and one in your aorta, they are sending a signal to your brain saying the blood pH is dropping. We're going to need to start upping our game in terms of gas exchange and delivery of oxygen to the tissues. And that basically does two things. It makes you breathe faster and it makes your heart rate go up. It does some other things as well, but those are the two big ones. Okay, so as the muscle tissue is respiring more, that is producing carbon dioxide. Then receptors detect high carbon dioxide concentration. They will then send a signal to your brain, and the brain then sends a signal to the heart telling it to beat faster. Okay, when the student first starts running, they respire aerobically. Write the word equation for aerobic respiration. This is straightforward knowledge. Here we go. Oxygen plus glucose gives you carbon dioxide plus water. And one advantage of aerobic respiration versus anaerobic respiration, there's a couple you could go with here. The biggest one is that you produce more ATP. So every molecule of glucose in anaerobic respiration gives you 2 ATP. Every molecule of glucose in aerobic respiration gives you between 36 and 38. So it's around 20 times more efficient to respire aerobically than anaerobically. That's the big one. I suspect you probably also get a mark in here for the lack of production of lactic acid in aerobic respiration, just because carbon dioxide is so much easier for your body to deal with. Lactic acid requires oxygen to get rid of it later, so it's borrowing against the oxygen that you will acquire in the future. So you can't just continuously respire anaerobically, even if you had a sort of unlimited supply of glucose. Okay, so we'll go with more ATP produced. You don't need to know the numbers for this, but just to put it in context, 2 versus 36 to 38 per glucose. The other one is no lactic acid production. This is a 1 mark question, so that's a sort of either or job. That's the whole thing. The mark scheme for this paper is at the back of your white CGP book. Do use that, it's pretty bare bones. I think it's important to be looking here in terms of model answers, probably more than mark scheme answers at this stage. But nonetheless, use that to assess your own and to be polishing as well. Thanks for sticking with me. Go well this term. I'll see you at mock time.